All right, guys, welcome back to Underground Science. In this video, we'll be talking about Chediak-Higashi syndrome. All right, so let's go and get started. So the basic overview, right, in terms of the genetics, this syndrome is autosomal recessive, all right, and it's actually the mutation in the list gene, all right? So it's a mutation in the list gene. Now, what is the list gene? It's the lysosomal trafficking regulator gene. All right, and it's going to regulate the transport of vesicles within the cell, the transport of vesicles, the transport of lysosomes, the transport of granules. All right, and remember, you need microtubules to transport like vesicles, lysosomes, and granules inside the cell. In Chediak-Higashi syndrome, this gene is mutated, right? And so that means that the microtubules fail to transport the vesicles, lysosomes, and granules inside the cell. Okay, so that's why we have here saying that in Chediak-Higashi syndrome, you have a defective intracellular trafficking of vesicles and, of course, also granules and lysosomes. So you can add that too of granules and lysosomes. Okay. Now, why is this again? Because remember, in this syndrome, the microtubules fail to transport the vesicles, lysosomes, and granules. Now, why is this bad? Right? This is this isn't good because it can affect certain cells. So remember, we said one was lysosomes, right? Lysosomes need to be transported inside of cells, and you do that via microtubules. But in this Chediak-Higashi syndrome, there's a defect in this system where you can't transport lysosomes across microtubules and inside the cell wherever it needs to be. Why is this bad? So let's look at neutrophils, for example, right? So let's say we have a neutrophil here. Remember when neutrophils see a pathogen, they phagocytose it, right? So this is our pathogen. The neutrophil will actually phagocytose this pathogen, right? So it's going to take it in. And when it takes it in, it's going to form a phagosome, right? So we're going to have our neutrophil here. We're not going to draw the nucleus for now. We're going to have our phagosome here, and then the pathogen inside of it. But remember, let's draw lysosomes. Let's say this is our lysosome, right? Remember, we need to take this pathogen and destroy it via the enzymes in the lysosome. So if this is our neutrophil or macrophage or whatever, right? If we need to take this and fuse it with the lysosome and form a phagolysosome, but to do that, you need microtubules and the proper function of those microtubules, right? To fuse these two together. But remember, we said it's a defect in the microtubules. So you can't really move things around in the cell properly, one of those being the lysosomes. So how are you going to move the lysosomes, right? How are you going to transport the lysosomes or inside the cell, and how are you going to fuse these two together, right? So that becomes a problem because then you can't kill this pathogen off properly. Okay, and we'll talk about what that leads to later, which is, of course, recurrent infections. So one thing we saw is in this list gene mutation, right, in Chediak-Higashi syndrome, the microtubule dependent vesicle trafficking is defective. And that affects the lysosomes too, because lysosomes need to be moved around inside the cell. For example, lysosomes need to be fused with the phagosome, where the pathogen is sitting inside of, in order to destroy the pathogen. If you can't do that, you increase risk of recurrent infections. Okay, so that's one example. Another example, if we take our melanosomes, right? So remember, we have 5 layers of our epidermis, right? Well, the stratum lucidum is in places where there's thick skin, like the palms and soles of the palms and soles, right? But if we look at the 5th layer of the epidermis, which is the stratum basal, right? Stratum basal. In the stratum basal, you have your melanocytes. So if we were to draw the melanocytes, let's draw it in this color. Let's say this is your melanocyte, right? And of course, we know melanocytes do what? Melanocytes make melanin, right? So let's say this is our melanocyte. and they're making melanin. So let's say this is our melanin, right? After melanocytes make the melanin, they need to package that into melanosomes. So let's say they do package it into melanosomes, and we're going to draw that in, I guess let's just draw it like this, right? So let's say these are our melanosomes, and inside the melanosomes, we have the packaged melanin. All right, so we need to now, in order to, for the next step to happen, you need functioning microtubules, right? So what needs to happen next is your melanosomes actually need to get transported from your melanocyte, okay, to the keratinocytes. So remember, your keratinocytes are your skin cells, and you need to transport the melanin, right, from your melanocyte to your keratinocytes. You need to distribute that to your keratinocytes. But in order to do that, you need functioning microtubules, okay, which you don't have in Chediak-Higashi syndrome. So that means these melanosomes just accumulate, right, inside your melanocytes, and it can't really reach your skin cells or your hair cells, and even your eyes, right? So we know there's pigmentation there too. So it's going to lead to partial albinism, right? Hypopigmentation. Okay, so so that's another effect. Okay, it's going to affect your melanosome transport. Another cell to focus on are your platelets. So if we have our platelet here, let's say this is a platelet, right? Remember, our platelets have dense granules in it. So let's say these are our dense granules. Remember, your dense granules release certain things in order to call more platelets and help with clotting. Okay, and we'll go over that when we talk about hemostasis and disorders relating to that. But you need these dense granules because, again, they have molecules that will help with clotting and calling more platelets to the area where you need to clot. All right. And if you don't, but in order for these granules to properly work, right, you need a proper microtubule function. But in Chediak-Higashi, again, remember, you have a defect. You have a microtubule dependent vesicle trafficking defect, right? There's a defect in this microtubule system. So that means these dense granules cannot be distributed properly inside the cell. So what happens is the dense granules actually accumulate, right, like this inside the platelets, and that's going to lead to platelet dysfunction, which can lead to bleeding. Okay, so that's talking about platelets. Another example would be your axons and your nerve cells, right? Remember, your nerve cells transport things to and from, right? From the cell body to through the axon and to the synapse and back, right? And that needs to happen in order for proper nerve function. You need to transport vesicles down to the synapse and back to the cell body, right? Across the axon. Normally, that needs to happen if you want good function of your nerves. Now, if that can't happen, because again, you have a defect and you have a microtubule dependent vesicle trafficking defect, right? And your microtubules aren't working properly, you can't really transport vesicles to and from in your nerve cells. So that's going to lead to nerve dysfunction. And we'll see what that means in a bit. Okay, so that's basic. That's the basic overview. Now let's talk about the findings. Okay, so the first finding we should talk about is increased risk of pyogenic infections, right? And this should make sense because we already talked about this, right? So remember, we said when neutrophils or macrophages phagocytose the pathogen, right? Remember, so let's say this is our macrophage or neutrophil or whatever, right? And it phagocytoses the pathogen and it has it inside the phagosome. So let's say that's the phagosome and the pathogen's like this inside. It needs to fuse this phagosome with the lysosomes inside of the cell, right? In order to destroy the pathogen. But if you can't do that because you have a defect, right, in moving granules around or moving lysosomes around inside your cell, right? Because you have a defect in your microtubules, and then you can't fuse your phagosomes with your lysosomes, then this pathogen won't be destroyed, which then makes sense to say that, okay, one of the findings is recurrent infections. Okay, because remember, you need to release, your neutrophils, for example, have granules in it that have certain enzymes that will destroy the pathogen. To degranulate, you can't even degranulate either, remember, because the granules build up and it can't move around properly, right? Your lysosomes can't move around properly. And you need all these in order to properly destroy the pathogen. If you can't, therefore the pathogen will survive and you're going to have an increased risk of infection, pyogenic infections, right? So it's actually going to be an increased risk of Staph aureus and Strep pyogenes. Okay, so which we have written here, Staph aureus and Strep pyogenes. All right. So pyogenic infections again means pus-forming bacteria that cause the infections. All right. So just the review in terms there. So this happens again because you have impaired phagolysosome formation due to the defect in vesicle trafficking and microtubules. So you can't really move around granules, you can't move around lysosomes properly, and you can't move around vesicles properly inside your cell. Okay. The second finding is neutropenia. Now, why would you get neutropenia? Well, we can talk about a few reasons, right? So one reason why you'd get neutropenia, so if we look at our neutrophils, we have our phagosome, right? And then inside the phagosome, we have the pathogen that the neutrophil phagocytosed. And then we have our lysosomes, right? So let's say we have our lysosomes like this. Now remember, our lysosomes have granules in it, toxic granules, right? That need to get fused with the phagosome. The granules need to get delivered. So these granules that we see here are inside the lysosomes. They need to get delivered to the phagosome in order to destroy the pathogen. But you have a microtubule defect, so you can't do that. So what happens is the granules build up, build up, build up inside the lysosomes to the point where it leads to the dysfunction of neutrophils, because then you can't really properly deliver the granules to the phagosome. Okay, so if the granules build up inside the lysosomes, they become so enlarged that you can't really have functional neutrophils because it's non-functional granules at this point because of how much it builds up because you have a defect in your microtubule, so you can't really transfer those granules to the phagosome. Okay, so that's one way that the neutrophils stop working. All right, and then that's gonna, of course, lead to recurrent infections. Okay, and because that leads to the neutrophils to stop working, you can see a decreased functional count of decreased count of neutrophils. All right. And what happens is these giant granules, when they build up like this, the neutrophils can undergo apoptosis also. So that can lead to low neutrophil count in the blood. Another way that you may see neutropenia, right, is remember where are neutrophils made, right? Neutrophils are made in the bone marrow and then they're released into circulation. But remember, you need microtubules, right, in order for the neutrophils to get or to go from the bone marrow to the bloodstream. And if you have defective microtubule function in Chediak-Higashi syndrome, then your neutrophils may get trapped in your bone marrow. Okay, and then that can also reduce the circulating levels of neutrophils. All right, so we have two ways. What we just discussed about the bone marrow, and the other way, which neutrophils, right? The granules build up inside the lysosomes to the point where the neutrophils become non-functional, and then this can lead to apoptosis of the neutrophils also, leading to neutropenia, okay, or low neutrophils in the blood. All right, so that's the reason for neutrophils. So again, we can see here that we listed one of the reasons. Neutrophils it won't be able to go from the bone marrow to the blood, right? Leading to low blood levels of neutrophils due to defective microtubule function, right? Defective vesicle trafficking. And so it may even get destroyed in the bone marrow also due to this reason because you're not able to get the neutrophils out from your bone marrow to your bloodstream. All right, so that's the reason for low neutrophils. Next finding, right, is we have giant cytoplasmic granules present inside leukocytes, like we already been discussing, right? So again, if you can't move around granules properly, then of course you're going to have a buildup of those granules leading to giant granules, right? So that's something we already discussed. Okay, so what we have here is if you can't move around vesicles or granules properly, right, your vesicles can start to fuse and clump together, right, leading to formation of giant granules, right? So if you can't move around certain granules properly or vesicles properly, they're going to obviously start to clump together, okay, because you can't move them around because of defective microtubules in Chediak-Higashi syndrome. Okay, so that's another finding that we've already discussed. Another finding to talk about is dysfunctional platelets due to the accumulation of abnormal dense granules, right? Remember, you need dense granules in your platelets to secrete certain molecules to help with clotting and to bring more platelets to the area where you need to clot. But because you can't move around these granules properly, you can't move these granules around in the platelets properly because you have dysfunctional microtubules, these dense granules, right, build up, and that can lead to dysfunctional platelets. So this leads to dysfunctional platelets because now you can't really release what you have in your granules, so you can't clot blood properly, right? Or you can't form that platelet plug properly. So that means you're going to have easy bruising and mucosal bleeding, okay? Because you have platelet dysfunction. So again, let's read what we have here, right? You're going to have a list gene mutation. So you have a defect in vesicle trafficking and microtubules that leads to accumulation of abnormal dense granules. Remember, something in your dense granules are molecules that you need in order to call more platelets to the area to help with the forming the platelet plug, one of those being ADP. So if you can't release these granules, one of them being ADP, then you can't have proper platelet aggregation because that's what ADP does, right? It helps platelet aggregation, bring more platelets to the area where you need to clot. And then therefore you can't form that platelet plug. So you're gonna have easy bruising and mucosal bleeding, okay, which is something you see in dysfunctional platelets, if you have dysfunctional platelets. Okay, so that's that finding. Another finding is albinism. Now, we already discussed this, right, in the overview slide. So if you have your melanocyte, right, like this, in your melanocyte, which we said is in the stratum basale layer, which is the fifth layer of the epidermis, right? If you're counting the stratum lucidum to be the second layer in thick skin like palms and soles, you have your melanocyte, right? Your melanocyte, we said, makes melanin, okay? And we said this melanin gets packaged inside of melanosomes. So if we draw our melanosomes like this, let's say these are our melanosomes, and inside our melanosomes, we said we make melanin, which will represent an orange. Then we said you need functioning microtubules to take this, to take these melanosomes and transport it to your hair, skin, to your hair, skin, and eyes, right? Like this happened in your eyes too, because your eyes need pigmentation also. So so of course, if you don't have functional microtubules, then you can't properly transport these melanosomes from your melanocyte to your skin cells, like your keratinocytes or your hair cells, right? So then if that happens, then melanosomes build up inside your melanocytes because you can't transport them, right? Because of defective microtubules. So if these melanosomes build up, so if we draw this, right? Melanosomes build up inside your melanocytes with all this melanin, and you can't distribute these melanosomes and melanin to your skin cells or your hair cells or your eyes, right? Then you're going to see hypopigmentation. You're going to see partial albinism, okay? Which makes sense now because of defective microtubule function, and the melanosomes build up, and you can't transport this melanin to other skin cells and hair cells, for example. And so then that will lead to hypopigmentation. Okay, so I hope that finding makes sense also. So you may see things like light, light colored skin, right? You may see silvery hair and light colored eyes, again, due to hypopigmentation because of the defect you have in your microtubules in this Chediak-Higashi syndrome. Another finding we should talk about is relating to neurological dysfunction, one of them being peripheral neuropathy. Okay, and let's see why. Remember, again, you need microtubules to transport vesicles, right, inside your cell. So in your neuron, you need microtubules, right? So if we draw a neuron like this, got our dendrites, right? Got your cell body, and then you got your axon, and then your terminal. Remember, we use dynein and kinesin, right, as our motor proteins to transport things to and from the cell body to the periphery, and from the periphery to the cell body. Your neurons need to do that also. They need to be transporting vesicles like this to and from in order to properly function. But if you can't transport these neuronal vesicles properly because you need microtubules to do this, then you're going to have neuronal dysfunction, right? One of them being peripheral neuropathy. Okay. And if you can't do this in your cranial nerves, right? If you have cranial nerves, like if this was a cranial nerve, for example, right? And you can't transport vesicles to and from, right? Then you're going to get cranial nerve palsies, right? Because you can't do proper axonal transport. So you can also see cranial nerve palsies too, if this is happening in your cranial nerves, right? So you may see peripheral neuropathy because you can't transport vesicles properly along the axon in peripheral nerves. You may see cranial nerve palsies if you can't transport vesicles across axons, right, properly due to microtubule dysfunction, right? If this is happening in your cerebellar cells, right, your cerebellar neurons, then of course you could see ataxia, right? You can see loss of coordination because that's what your cerebellum is important for. So those are the main things that we would find, right, relating to the nervous system. Ataxia, you would see peripheral neuropathy, right? And you can also see cranial nerve palsies too. And again, that's because your microtubules don't work properly, right? So you can't transport vesicles along your axon. That can lead to neuronal dysfunction, okay, or nerve dysfunction. All right, so those are actually the main findings um, of Chediak-Higashi syndrome. Okay, so I hope this video wasn't too confusing. I hope this made a little bit of sense. And thank you guys for watching. See you guys in my future videos.